So it's my great pleasure to introduce Dr. Duojia Pan as our keynote speaker of our symposium. So DJ is currently the Basho Distinguished Professor and Chairman of the Department of Pathology at the University of Texas Southwestern Medical Center. DJ got his bachelor degree from Beijing University. So we're both PKU alumni. And he came to the US and did his PhD study at UCLA School of Medicine, and then his postdoc training with Dr. Jared Rubin at UC Berkeley. He started his lab at UT Southwestern in 1998 and stayed there till 2004. That's when I got to know him because I was doing my postdoc during that time. And we both work on fly. It also reminds me, my first time reading his milestone paper, 2003, that Cell paper, who discovered Hippo pathway, such an elegant science, such important finding. And in 2004, DJ moved his lab to Johns Hopkins University School of Medicine, together with wife Elizabeth Chen, which many of us also know. So they stayed in the area till 2016. During this time, his lab discovered the upstream regulators of the Hippo pathway and further established conserved roles of Hippo pathway in mammalian tissues for the mammalian tissue growth, regeneration, and tumorigenesis, and also CKD. And in 2016, he accepted the chairman position at the Department of Physiology at UT Southwestern and moved back to Dallas. So his lab published over 70 papers on very high impact journals, and he has very high standard on publications and patents, and he also received many awards, include the Paul Mars Prize for Cancer Research, and he has been a HHMI investigator since 2008. So today, if you search Hippo pathway on PubMed, you'll see over 5,000 papers. All of them originate from DJ's original work, discovering this very important signaling pathway. So without any further ado, let's hear about this fascinating work from DJ.
Thank you very much, and for that very kind introduction. I would also like to take this opportunity to thank Jay and Hong and Yinxi for organizing this wonderful event. It's especially nice for me to see so many old friends here, although virtually, but also to see some of the new sciences and meet some new friends today and listen to some of the very, very beautiful talks and wonderful images, especially I've seen today. So it's a really lively community in Baltimore and Washington area, because I know today I still miss my friends there and all the restaurants I often go to and the friendship, and so will always be with me. So what I like to do today is to kind of give you an overview of the Hippo pathway and its role in growth control, but also to really take you to a more kind of a speculative thinking about how this pathway comes about during evolution. And so because it's getting end of the day, so I like to, everybody just relax, because I'm just going to give you a very kind of a general talk and without giving too much data, and just trying to kind of present the big picture and rather than dig into the details, okay? And so, so today I will, as I just said, to first give you an overview, I'm going to tell you something about the evolution, okay? And so the whole discovery, the whole Hippo pathway can be traced back to about 20 years ago, when I was about to finish my postdoctoral work with Jerry Rubin. Because in Jerry's lab at that time, they developed a very powerful technique that allow one to generate what we call a genetic mosaic in a very efficient way. The idea of genetic mosaic has been there, in genetics for over a century, but it's only, around that time that people actually developed a very high, a very sophisticated technology to do it efficiently, such that you can even use such a technology to do unbiased genetic screen. So I won't want to get into the details of these screens, how they are done, but the general concept is very simple. That is, you can take a fly, in this case, a male, and mutagenize with any kind of chemical mutagen. And then you can cross this male fly with, of course, unmutated female. And in the next generation, because of these genetic strains, the strains are genetically engineered such that they can allow you to generate very high frequency genetic mosaic, in this case, in particular in the fly eye. And the beauty of the system is that the mutagenized chromosome and unmutagenized chromosomes are marked differentially by eye color. So basically, when you get a mosaic eye, which composes of both white color and red color, the ratio of the two color basically give you a representation of the relative growth potential of the two population of cells. So in this just the example. So normally, let's say you have a control cell and it's unmutagenized, but and let's say it's marked by the white color, and the white type, this is also a white type cell, but they're marked by red color. You can see they have certain ratio. However, in this particular case, we have a mutant chromosome, which is again marked by a white color, but when it's next to these red color, the white type cell, you can see that there's an overrepresentation of mutant tissue, which are marked by a white color. So it's a very simple visual screen anybody can do. And so, and besides, screening, of course, we do it in the adult eye, you can also go back to development. In this way, we often actually use a positive marker such as GFP to mark the mutant clone. So in this case, you can see that 3 separate clones in a developing tissue during novel development in the fly. And you can see normally these clones have certain size and a number of cells in each clone. However, when you are mutating a specific gene and you actually have these clones growing very big, and often actually these overgrown clones are very, very long in terms of their shape, okay? And so these, again, provide a very powerful, we can start to look at it like gene expression during development and in these mutant cells, specifically what happened in these mutant cells as compared to their GFP negative neighboring Y-type cells, right? So it's a very simple approach. So using this approach, I always go back to my scientific training, and this is actually something written by my postdoctoral advisor, Jerry Rubin, which has just stuck with me for the rest of my career, which I thought is very, very powerful. And that really speaks to the power of fly genetic. And I just read it here. That's basically says the flies are particularly amenable to a black box approach in science, studying the consequences of manipulating genes and cells using clever assays that can yield information that does not depend on having a good hypothesis. You can gather information in a non-biased way on how things work. So I think this concept has been sticking with me for my whole career, because whenever we study a new question, we just say, let's do a screen, all right? So I think that is an approach I really, personally favor. So from this kind of screen, when I started in my lab as initially at UT Southwestern, I started doing such a screen and look for, tumor suppressor gene. And interestingly, at that time, I'm still puzzled by why these genes haven't been discovered before us, because, by the time I started my lab in 2004, or started 1998, this technology was there for about 5 years already. So, many people have been doing such a screen. And so we were lucky, actually. There are still a lot of genes to be discovered using this approach. And we basically did such a screen. We uncovered 2 classes of mutations that can increase like clone size. And in the first class of mutation, you generate this increased clone size due to an increase in cell size. So the cell numbers are pretty much not unchanged, but the size of each individual cell become larger. So these class, the first class includes 3 particular genes, and which actually we studied very extensively. In particular, you can recognize these genes now because, P10, of course, was a very famous tumor suppressor. But at that time, TSA1, TSA2 were very new. Actually, nobody knew much about their biochemical function. Actually, our work, and along with the work from several other labs, established that TSA1 and 2 actually functions as upstream regulator of the mTOR pathway by directly functioning as a GAP, GTPase activating protein for the small GTPase Reb, basically to inactivate Reb, as you know now today, of course, that Reb is actually a very direct activator of mTOR. So this really especially the power of fly genetics that allow you to really, really fishing out really this critical upstream regulator of the mTOR pathway. But while we were doing this work with the cell size mutant, we also got a second class of mutations, which their phenotype are much more dramatic than these. I mean, here is an example of a TSA1 mosaic eye, where you can see that this part of the eye is, represent mutant tissue, but this part of the eye represent wild type tissue. You can see that these guys, these cells are bigger, right? But we have another class of mutation, which is very dramatic. This is what you get. Basically, the eyes are completely screwed up. They have these multiple folds. And this phenotype not just restricted to the eye. If you were to make mutant clones in other parts of the body, for example, on the fly thorax, you also generate these massive, tumors. And so these phenotypes were very striking and very, indicative of something, maybe a pathway. And fortunately, at that time, actually the number of genes is not just one gene. There are multiple mutations that were isolated, including Hippo and SAB and Wilson and MAT. So there are 4 genes that all give the same phenotype. So it's clear to any geneticist that these genes may form a new pathway. And lo and behold, actually, the work from our lab and several actually characterizing these mutations demonstrate indeed these proteins form a new segment cascade. And I would just like to highlight some developmental characterization of these phenotypes, tell you why these particular pathway give you such a dramatic tumor-like phenotype, which is much more dramatic than, say, for example, perturbing the mTOR pathway, for example, right? I think what really happened here when you perturb the Hippo pathway, again, here we use mosaic analysis, we can unambiguously label Hippo mutant cells by the lack of GFP marker. Okay, so whenever you say the GFP negative, those are the Hippo mutant cells. You can see here, when you look at cell proliferation by looking at phosphorylated histone 3, at this time of development, normally cells already exit the cell cycle. But in the Hippo mutant portion, in the mosaic, these cells, they continue to proliferate as if, they are still divided. So they fail to exit cell cycle at the time when they are supposed to exit cell cycle. Conversely, if you were to look at the apoptosis, again, at this point of time in development, normally cells, the fly generate more cells than necessary to build a fly eye. And these extra cells are purged by developmentally programmed cell death, which are shown here by the tunnel stem. However, in the mutant portion of the mosaic, you can see that in the Hippo mutant cell, they do not undergo any developmentally programmed cell death. So this really dramatic phenotype is due to a combined effect of a failure to exit cell cycle, as well as a failure to execute developmentally programmed cell death. And then actually, even to be more dramatic than this, we can actually show that in these Hippo mutant cells, you can even give them very, very high dosage of gamma ray radiation, which normally would kill 99% of cells in the fly, development tissue. And in these mutant tissue, 0 cell die, actually. So these are extremely, extremely powerful phenotype. And so anyway, we were, of course, very interested in understanding what happened, what's under, why this pathway is so, powerful. And to make a long story short, we were able to show that these 4 tumors, what we call tumor suppressor protein, because now the function of any of these genes leads to a very powerful tumor phenotype. They all function through a single molecule, okay, in fly, which is called YAP. And the YAP is what we call a transcription co-activator. So normally it can go to the nucleus and function with a DNA binding transcription factor called the scallop to turn on genes to drive cell proliferation, to suppress apoptosis. So it's basically a growth promoting gene. But during development, what happened is that the YAP gene, has to be somehow turned off by the Hippo pathway, by these tumor suppressors. So these proteins form a kinase cascade. I didn't tell you what these proteins are, right? The Hippo is actually protein kinase. It phosphorylates a downstream kinase called WRTS. And this WRTS protein then phosphorylates this YAP transcription co-activator. So once YAP is phosphorylated, it can no longer go into the nucleus, stays in the cytoplasm. That's how it's inactivated. We think the whole pathway have to go through this YAP transcription factor because you can simply activate the YAP, and that genetically can completely phenocopy anything you see with loss of function of any of these upstream tumor suppressor. we can actually even go to the specific phosphorylation site within YAP and mutate a single phosphorylation site to show that that one point mutation in YAP, okay, can result in the complete phenocopying of this any of the loss of function phenotype for the upstream tumor surface. So it's clear now that YAP, it's the only output of this pathway as far as growth control is concerned, okay? And so this here just show you some of the phenotype, which I already showed you the Hippo loss of function phenotype. You can see the very dramatic tumor phenotype. And this is what happened, if you simply activate YAP, you can generate huge organs or big clones, okay, in a developing tissue. And so while we are doing this work, right, and like as Jay mentioned, we're very happy to work on these genes in the fly. We immediately realized that all of these genes are conserved in the mammalian system. So that begs the obvious question to anybody, that is whether this pathway is conserved in mammal, and if so, what do they do? What does it do? And so we were very lucky because as a geneticist, we examined these kind of conservation of genes between fly and mammal. We immediately realized that, because YAP is really the output of the pathway in flies, the prediction would be, if you were to look at the mammalian counterpart of YAP, which include the two orthologs, which is called YAP or TAZ. These proteins should also be phosphorylated, right, by their mammalian counterpart. Indeed, we were able to establish that by kinase assay. But more importantly, we made a prediction based on the genetics that we learned in fly. That is, if you were to simply overexpress the mammalian counterpart of YAP, right, in a mammalian tissue, you should, in principle, generate a huge tissue overgrowth, as what we saw in Drosophila, right? So that's actually the first mouse model ever generated in my lab. And so we actually went on to generate a transgenic tetracycline inducible YAP model, in which we specifically turn on this transgene in the mouse liver, and using this tetracycline inducible system. As you can see that, when you turn on this gene, within a few weeks, the liver grow to 4 or 5 times of its normal size. This is what happens if you induce in adult. But if you induce this overexpression for extended period of time, let's say after 2 months of time, you start to see these very dramatic tumors. And so, let me see what's going on. So you start to see these tumors. And these individual foci, actually, we look at them histologically, they're really what we call the hepatocellular carcinoma, okay? And these are what happen, if you induce in the adult stage. But if you were to induce YAP transgene, at the birth, right, you can do so by simply feeding tetracycline to the nursing mother who are still nursing the pup, right? And from the milk, the pup will get tetracycline and turn on the transgene in their liver. If you do so, you can see that within a month, that the pup's liver is completely covered by hepatocellular carcinoma. So this is extremely powerful, potent phenotype. And so, so this immediately told us this pathway must be very important in cancer. And so actually very interestingly, nature actually has done the overexpression experiment for us in human. And instead of we doing all this fancy experiment in flies or mice, nature has done it already in human. Because, and we know that actually the YAP and the TAZ gene are known to be amplified in many different human cancers. And so this is some of those examples here, such as lung cancer or ovarian cancer. At certain frequency, these genes are known to be amplified. But as any of you might know who work on cancer, when you ever have an amplicon in a human cancer, typically the amplicon contains dozens of genes. It was actually not easy to tell which one of these dozens of genes represents the real driver within that amplicon, right? So here's actually the example of the YAP amplicon, which is present on chromosome 11. And this amplicon is well known, but it contains dozens of genes. For many years, people were suspecting some other genes in this interval were the driver, right? That's responsible for this amplicon, phenotype in human cancer. For example, here we know YAP is here, but it also has many other familiar genes. So people actually publish many papers like here. So CIP1 or CIP2 is a driver. But of course, that's all wrong. And now we know clearly it's YAP, within this locus. And another way, actually, again, nature played with us about telling us about YAP, the importance of YAP and TAZ. It's through gene fusion. And so you can see that actually both YAP and TAZ are known to be present in certain gene fusion that result from chromosomal translocation. And so these YAP translocations, this nomenclature basically means that YAP is somehow fused with another gene, and here's a YAP fused with another gene, and here is a TAZ fused with another gene called CAMTA1. So these are just different chromosomal translocations that result in the constitutive activity of these transcriptional co-activator. Right? And so actually one particular example I'd like to share with you is this very rare vascular tumor in human, which is called EHE. Okay, so these EHE tumors are extremely rare, but almost in every single sample that people were able to look at this EHE, it absolutely contains this gene fusion called the TAZ-CAMTA1. So basically it's a fusion between two genes, TAZ, which is a factor of the Hippo pathway, and another transcription factor called CAMTA1. And this is actually due to a chromosomal translocation. You can see normally, you can see that these guys have a little extra stuff on chromosome 1, and the last a little bit stuff on the chromosome 3, right? This is the translocation that lead to this fusion protein. And we were able to show that actually this fusion protein, just this one fusion protein alone is sufficient to completely mimic the EHE phenotype, by inducing this vascular tumor. And furthermore, if you were to silence, once you induce this tumor, if you were to silence the TAZ-CAMTA gene, and then this tumor then just disappeared, right? So this basically tells you that this particular fusion gene is not just a bystander passenger; it's actually a driver for this particular type of cancer. And so, more broadly now, actually, I'm citing a paper published by others, as in Cell about a few years ago, where they actually look at comprehensively among all the different cancer types cancers in the TCGA database, and look at all the different pathways. And actually, Hippo is one of the major pathways now we recognize that's mutated in human cancer, along with other pathways such as RTK, RAS pathway, PI3K, and March and Wind, and etc. So it's one of the major pathways. And in this particular study, they were not only looking at the YAP and TAZ mutation, right, or amplification, they're of course also looking at the mutation, loss of function mutation of the upstream tumor suppressor genes, okay? And so obviously there's a lot of interest in trying to, target this pathway for cancer. And about 10 years ago, in collaboration with Jun Liu's lab, we actually screened his very famous FDA-approved drug library. We were able to identify a small molecule called verteporfin, which can actually block the activity of YAP by inhibiting this physical interaction between YAP and TAZ and its DNA binding transcription factor called TAD. So here are just some examples. You can see that when you increase the increasing concentration of verteporfin, you can actually decrease the physical association between TAD and YAP. And that actually allowed, not only you to disrupt this conflict, but it actually has effect on this transgenic mouse model I just showed you in the liver overgrowth model. If you treat it with verteporfin, you can actually suppress the liver growth. And actually many labs are now using this compound. It's a tool compound. This compound still have some other issue, but it's at least a good tool compound and to look at the YAP dependency, in an experimental setting. And so this is just some example of work from other labs which use verteporfin to show the YAP dependency on a particular type of tumor called the uveal melanoma, which they have shown is dependent on YAP activation. And they can show that with very repeat treatment, you can significantly suppress these kind of tumor. So far, I've told you about a lot of stuff. I told you about how YAP, right, is important in the tumor cell, right? It's a driver of tumor cell proliferation, for example. But another collaboration with my colleague at Johns Hopkins, we actually several years ago we discovered another layer of about YAP and TAZ and the Hippo pathway that's also very interesting. In this particular story, which we did together with Fan Pan and Zhu Padel's lab at Johns Hopkins, we were not looking at the tumor cell. Rather, we are looking at the immune system. We are looking at how, specifically in this case, how YAP functions in the particular type of T cell called Treg cell. And as many of you know, Treg cells are essential for self-tolerance and immune homeostasis, because it allows us to tolerate certain self-antigen. But because of this immune suppressive activity, it actually also bad, right, for anti-tumor immunity, because the Treg cell will suppress our body's intrinsic ability to fight against cancer, right? And so it turns out actually YAP is highly expressed, in the Treg cells, okay? And we actually ask the question, what is doing, right? What is YAP doing in these Treg cells, which normally suppress anti-tumor immunity? So we were able to make a T cell specific knockout of YAP in the T cells only, right? And then we transplant, melanoma cells into these, immune competent mice, right? But in the normal mice, of course, these melanoma cells, they will grow very big, right? However, in the mouse in which we knock out YAP specifically in the T cell lineage, and these tumor cannot grow at all. So this actually tells you that the YAP, protein is not only driving the growth of the tumor cells, right? It's also playing a very important role in suppressing the anti-tumor immunity. So in other words, we envision that the YAP inhibitors such as VP, and also many actually companies are now developing YAP inhibitor, can be serving as a dual agent that can not only target the tumor cells per se, but also improve the anti-tumor immunity from the immune system. So I think this is kind of hold a good promise, for future application of these inhibitors. So far, I've told you a lot about, YAP and TAZ, how it's control growth, promote growth, how it's important in cancer, because it drives a lot of feature of cancer cells. But there's another side of the same coin, that is, YAP and TAZ, they actually also do some very good, important role during normal physiology and normal development, especially for regeneration. And it turns out, actually, in almost every experimental model people have looked at that involves any kind of tissue injury or tissue regeneration, whether you do it in mouse or zebrafish or whatever, system, YAP and TAZ always activated and play an essential role. And actually, one of the first demonstration of this came from our studies of an intestinal regeneration model in which we use DSS, which probably many of you have heard about, dextran sodium sulfate, which is a very commonly used experimental procedure to induce damage to the colon, and then allow the colon to self repair and regenerate. And so we noted that in this very commonly used regeneration model, you see a massive upregulation of YAP protein at a post-transcriptional level. Actually, it's not transcription, it's a post-transcriptional level during the regeneration phase. So basically what you do is you treat these mice with DSS for 5 days, then you withdraw DSS and let the colon repair itself. So during the repair, you see have a time window where YAP expression is extremely high. And we show that this expression of YAP during this repair phase is essential for regeneration, because if you were to knock out the YAP gene during, in the adult, and they don't have any phenotype, right, on the normal homeostasis, normal condition, but only when you treat these mice with DSS, now these mice, they cannot repair, and they basically die. Okay, they cannot deal with the injury, they cannot repair, and they just fail. And so this actually become a common theme in, as I said, in all regeneration model. And I would just share with you the work from my colleague, which Jay is very familiar with because he did his postdoc work with Eric Olsen. And also James Martin was a former postdoc from Eric's lab. And so both labs actually showed that the Hippo pathway and the YAP actually is essential regulator for heart regeneration. So with the same direction, that is, if you decrease YAP activity, you compromise regeneration. And if you increase YAP activity, you actually improve regeneration. So in other words, I think there's also a lot of interest in trying to develop YAP activators that can be utilized to benefit regenerative medicine. So basically, we think about YAP and TAZ as like, a coin with two sides, right? On one side, we want to, inhibit its activity for cancer, but on the other hand, you might want to promote its activity, at least transiently, to improve regeneration. Okay, so far I have given you a very simplified view of the Hippo pathway, in which I concentrated on this, what I call the core kinase cascade, which is really go from the Hippo kinase to the Works kinase. And these other proteins, SAB and MAT, are basically their regulatory subunit of these kinases, okay? And then converge on transcription machinery. I did not want to give you, kind of a more complicated view, which a lot of people are now working on, including us. That is, what is upstream of this kinase cascade? So I think this is becoming less clear, but at least what we can say is that a lot of genes have now been shown to regulate this pathway. Is that here I will just show you some of them, which we and others have published. And so these are cytosolic proteins or membrane-bound proteins that can somehow regulate the Hippo pathway. I certainly don't want to bore you with the detail. But what is perhaps one of the most interesting features of these upstream signal came from the fact that, even the active cytoskeleton and the spectrum cytoskeleton, they actually are profound important regulator of the Hippo pathway. And this actually led to this idea that the Hippo pathway is not its main role is perhaps not, unlike the canonical pathway that you might be more familiar with, like Notch or Wind or Hedgehog or TGF-beta, which are normally utilized to respond to chemical ligand, right, on a cell surface or secreted ligand. And the Hippo pathway might be more important mechanotransducer, a pathway that's utilized to sense the mechanical property of the cell and the tissue architecture of the cell. And so here, actually, I just want to show you, some of the one little movie that we actually were a system we established in our lab to try to study this question about how the Hippo pathway responds to mechanical regulation. So in this particular case, we actually established the ex vivo system in which we can culture the developing fly tissue, ex vivo, and then apply mechanical force, and then ask question, what happened to the Hippo pathway? And so here's a movie in which now we culture this. Now we're looking at YAP, which is a nuclear, right? The nuclear effect of the pathway. So normally at this time of the development, YAP is phosphorylated by the Hippo pathway, it's inactive. That's why it's in the cytosol. So these cells have very, very small cytosol. And that's why you have these big holes, because each of these holes are the nuclei of the cell, right? So here I want to play this movie, and you can see that once you apply mechanical force, and YAP immediately go to the nucleus. Okay, so, and I can show you this again. So once you apply the force, YAP very quickly goes in. So this actually provides a powerful tool for us to dissect the whole mechanotransduction pathway upstream of it, right? Because you can generate a clone, for example, of any gene of interest and ask question, what happened to YAP nuclear translocation in that particular mutant clone in this mosaic tissue? So that's a story that we are trying to finish now. But I'd like to kind of give you a more broad view here, is because the Hippo pathway actually does many more things than just growth control. Here I just show you, so far I told you about growth, but actually, Hippo does a lot of things which may not have anything to do with growth control. Actually, during early development in, say, mammalian embryo, like mouse or human, actually, a Hippo pathway actually is required for the very first cell fate determination event in the blastocyst, which is the specification of the so-called inner cell mass versus the trophy ectoderm. So this, by bipartite cell fate determination absolutely require Hippo. So if you remove the Hippo pathway, and now all the cells differentiate as the same, now the inner cell mass also differentiate as the trophy ectoderm cells, okay? And there are some other paths, and also actually several years ago we published about the Hippo pathway involved in innate immune response. Again, I don't want to get into the details here, but the idea here, the Hippo pathway, but a lot of more things. But the key thing here is that this core kinase cascade is absolutely conserved, and it can be utilized to sense many different conditions, either during development or in stress or in physiology. Okay. Now I'd like to really tell you some kind of unpublished work, and again, kind of more speculative about how the Hippo pathway comes about and what it's doing and during evolution. Okay. So we were interested in this question for a very simple reason, because we initially worked on the Hippo pathway and we discovered all these components. We always wondered about, where this pathway come about during development. And this is actually an evolving story for the field, because very early on, many people actually, this just, I give you one example here, but the other people have published very similar paper to say that Hippo pathway is only present in multicellular organism, right? That makes sense, because as I already showed you, Hippo pathway controls cell proliferation, organ size, and it makes sense that it's present in multicellular life. And actually, these people actually were able to do phylogenetic analysis, looking at the genome of different species, at the different level of evolutionary tree, and show that, for example, the YAP, right, which is the most important target of the pathway, only evolved in what they call the, after the appearance of peripherin. So these are like a sponge, right? So they actually show this, they only, present after sponge. And so this is because at this time, about 10 years ago, many genomes were not sequenced, right? So that's the data. And it turns out actually around this time, the Broad Institute has been sequencing a lot of what they call unicellular relatives of metazoans. So these organisms have a very special position during evolution. They are single cell organisms. However, they're much more closer to, to the multicellular life, and then see that where like to plants or or like Neuraspora, for example. So we once these genomes were sequenced, it became clear to us. Actually, this is a collaboration between my lab and the Inaki Reis-Trino's lab in Spain. When we look at these genomes, we noticed that actually Hippo pathway was present already in some of these unicellular, higher unicellular organisms. In particular, we're interested in this one organism called the Capsospora, which belongs to a class of organism called the phylosterin, and also this other organism called coYki-regulated, which probably is more famous. Many of you have heard about it. And they all have, they are single cell organism, but they have the whole Hippo pathway, especially, the YAP homolog or the YAP, homolog. And so we were very interested in what this pathway might be doing. But just give you a kind of relative, a big picture here. This actually tells us the Hippo pathway is a very, very ancient pathway. And for example, if you were to put the Hippo pathway on the evolutionary tree, you see that Hippo appear here, okay, before, right, at the level of this phylosterin and coYki-regulated. But large TGF-beta Wnt do not appear, until at the level of sponge, okay, and hedgehog is even later. So Hippo is actually probably one of the most ancient pathways among all the canonical major signaling pathways. So we were very interested in understanding, where Hippo pathway came about. And so we did a very simple experiment at that time, right? That's like almost 10 years ago when we saw the genome. And we took the Capsospora homolog of YAP and the scallop, right? So these are the transcription effector of the pathway, right? And we did a simple experiment. So we simply overexpressed the Capsospora homolog of scallop and Capsospora homolog of YAP in the fly eye. And you can see that these eyes grow huge. So this phenotype is really identical to what you will see if you were to overexpress the fly version of these same genes, okay? So this tells you that, even though these organisms are so far apart during evolution, somehow these transcription factors still retain the transcriptional specificity to recognize, right, their targets, the same target as in flies, as their fly counterpart. Of course, the big question to us is, what is this pathway doing in this organism itself? And so this is when a very talented postdoc, Jonathan Phillip, came to my lab. So at that time, I was still at Johns Hopkins. So he wrote a letter and to me and say, hey, I want to come to your lab and study these organisms. So Jonathan is a perfect person to study this because he was working on dictyostelium as a graduate student. He knows a lot about these, simple unicellular organism. And so he was very intrigued by, our discovery of YAP and Hippo pathway in Capsulospora. By the way, I can tell you that dictys, for example, does not have Hippo pathway. So he feels like this is amazing. You have a single cell organism which have Hippo. So he came to my lab and he started to develop everything that you need to answer this question of what is the function of this pathway in Hippo, in this organism. And so before I go there, I just want to kind of give you a very quick, introduction about this organism, Capsulospora, because many of you, I'm sure, probably all of you haven't heard about it. So this organism is actually a very, very relevant, a very important organism, even from a medical point of view. And I'm sure all of you have heard about schistosomiosis, so which is a very important tropical disease. I think the Chinese for this is 血吸虫病. I think that's perhaps the Chinese of that. So these organisms, they have to go through two hosts, right? One, of course, is human, right, where they cause the disease. But they also have to go through what they call an intermediate host, which is snail, freshwater snail. So basically, the schistosome, they have to, when they grow in human and cause the trouble, then they get excreted through, from their poop, and they go into the water, and then they will have to go inside the snail to go through a specific stage of development. Okay, and then they get liberated from the snail, and then in the water they can, come back and infect people, right? From their water source, okay, contaminated water source. And so what is interesting is that this Capsulospora was initially discovered in such snail, which happen to be the host for schistosome, intermediate host for schistosome. And so it turns out that whenever they see this Capsulospora organism, if a snail harbors Capsulospora, then the snail cannot propagate schistosome. So because the Capsulospora amoeba, they actually attack schistosome sporocyst to kill these guys. So this kind of self-cleaning mechanism, this kind of a very complicated symbiotic mechanism in which, schistosome try to utilize snail to propagate, but then Capsulospora or, hijack will kind of kill these schistosome, sporocysts and to prevent that. So this is actually very interesting ecology going on. But in any case, so we took this organism and started growing in our lab, and we noticed something very interesting. That is, if you were to grow these in a regular plastic plate, which many of you know, they are very charged, okay? So these tend to be more sticky, and then they will just grow like a single cells, okay? However, when we grow these guys in a no adherence place, and then we start to see these clusters or aggregates. And so that's very interesting. And I think that's basically says these organisms, although they are single cells, they somehow have some kind of social behavior. And so Jonathan were able to look at these organisms and say, what happened in this aggregate, right? He was able to develop techniques that allow him to look at, let's say, actin, he can label actin, he can do EDU incorporation to cell proliferation. And here's an example you can see that these guys make a very extensive actin network, and they actually even make actin-rich protrusions that collect each cell in the cells in the cluster. But what is very interesting about EDU is in the EDU level, in my view, if you look at these clusters, this aggregate, you do not see any pattern, right, of EDU incorporation, which means the cell proliferation is distributed uniformly in this aggregate. And this is very different from what you might normally see in a tissue culture of mammalian cell. For those of you who look at organoid or mammalian cell culture, you would know that there's a very important phenomenon in cell culture of mammalian cell, which is contact inhibition. So basically, often when you see that the cells at the periphery of a cluster or a field of cells, these cells are more proliferative than cells in the middle because these cells are not undergoing contact inhibition, they're not being contacted by their neighbors, right? But in our case, when we see these clusters, you do not see any signs of contact inhibition. And furthermore, we can, these cells tend to be, they can merge with each other. In this case, we form two different clusters, all right? One not labeled with red, one labeled with green. We put them together, they can intermingle, okay? And they mix with each other. So these cells, they clearly have some, very interesting social behavior. And I also know that the calcium is important because we can utilize calcium chelators such as DGTA. And if you treat this with DGTA, these clusters completely dissociate, okay? And if we can add more calcium, of course, you can prevent the action of this calcium chelator. But the big question still come back to this question I told you about, what is Hippo doing in this organism? So Jonathan were able to develop, spend a lot of time to develop two technology. One is to be able to generate stable overexpression in these organisms. So before him, people were able to do some transient overexpression, but never stable overexpression. So he was able to generate a stable overexpression cell line of overexpressing any genes. In this particular case, we use a gene that's M scarlet just to be able to label the cells with color. You can see that in this population, we can generate a single cell clone, and so that every cell would have the same transgene, right? So they all labeled by M scarlet. But perhaps more importantly, Jonathan spent 5 years developing knockout technologies, and I can tell you, we tried everything under the sun, including CRISPR, of course, including RNAi, tailing. None of these worked. And actually, what did work for us is the classic homologous recombination. So for those of you who knows about homologous recombination, this is actually very similar to what you normally do in a ES cell culture of mouse. When we are generating a mouse knockout, you use this kind of, design of homologous recombination. So basically, we can have a homologous arm, two homologous arm, to an endogenous locus, but in the middle we put a, antibiotic selection marker, so that allows us to do selection. So using this technology, he was able to generate a knockout, a first knockout ever in this organism. Actually, the first knockout of any genes in these what we call the close relative of multicellular life, actually. And so he asked the question, what is this YAP gene doing in this organism? Surprisingly, actually, he tried very, very hard. There's absolutely no phenotype in cell proliferation. So this is what's surprising, because we know either the mammalian or fly counterpart of this gene is so important for cell proliferation. It has such a potent, proliferative effect. But if you were to look at the YAP mutant in this organism, either you grow them on a solid substrate or you grow them in shaking culture as well, we could not see any phenotype. And also we would look at the proliferation in this aggregate. Remember I told you they can, on the certain condition, form these aggregate, but even in this aggregate with the EDU labeling, we do not see any phenotype, okay? But we saw some other very interesting phenotype. So here's the phenotype. We noticed that if you look at the Y type, right, these guys form these aggregate, they're always very round. They form like a round ball, okay? This is like versa. But for these YAP mutant cell, they often form these aggregate that have very irregular shape. So if you were to measure what we call circularity, you can see that these are much more circular than these guys, okay? And actually, I will show you some movie here. And so to show you how we can watch the whole process of aggregation, and you can appreciate why these guys do not form these round structures. Okay, so I will start playing this movie. So here's left is Y type, right side is mutant. You can see that these cells initially, of course, they're separated, they start to aggregate together. to form these aggregate, right? In the wild type, both in the wild type and the mutant. But I hope you can appreciate that the wild type clusters are always very round, but the mutant one, they have a very irregular shape. And although they can merge together, but they are unstable. They can even split and undergo fission. Okay, so you will see some examples of fission only in the mutant side, but not on this wild type side. You can see here's what was a fission example. So they can spin off some cells out like here, right? You saw this one. They just split in the middle. So basically, these clusters are unstable, right? They are irregular shape, and they can fuse, but they can also undergo fission. So that's the idea. And so we, I think and another phenotype we observed in this guy is at the z-axis. Remember that movie we saw? Is really looking at like a 2D view. You look from the top, right? Look at XY axis, and you can see that the one is round, one is not round, right? So this actually, I really enjoy this movie. I always like to play this movie. But anyway, so you get the point. I will just quickly zoom into here. So we'll just end here. So anyway, you get it. So why you probably already noticed from this video that the wild type cluster, they're very black. Actually, it's because they have higher z-axis. They're taller. Okay, they're really like a round ball. But these guys is more kind of transparent because they are flatter, and which is very vividly shown in the next slide. So here we are trying to look at the z-axis, right, by looking at the side view. You can see that here's the substrate, right? Here's the plate surface, and you can see that these guys form a round kind of ball, sitting on the plate. But these other guys, the mutant, actually, they're kind of flat, okay? They do not form this 3D structure. So here's kind of illustration of what I'm trying to tell you. So one is a round ball, one is more kind of flat, pancake, right? So the question is why, right? When you see this phenotype, and intuitively you can think about two models, right? To explain why something will look like this versus this, right? So when you see a 3D structure like this, becoming something like this, either you can think about two scenario or a combination of them, right? Either like they say you have reduced the cell-cell interaction, or enhanced cell-substrate interaction, right? That could also lead to this kind of shape, right? Because if you somehow think about the cells that adhere to the surface more, right? Then they may promote this kind of spreading, or your cells stick to each other less. That may also allow you to see this. To make a long story short, which I don't want to bore you with this, so basically we see no evidence of cells stick to each other less in the mutant. So here just one example in which we look at the differential sorting, right? So we can, put wild type cells and mutant cells label different color and show that they actually, they intermingle, they do not sort out. This is very different, let's say, from the classic example where cell adhesion molecules, for example, different strength of cell adhesion will lead to automatic cell sorting. So these cells, they seem to sort each, just like wild type cells, But they do have very profound phenotype in cell substrate interaction. Actually, these mutant cells, they stick to the plate much stronger. So in this case, what we did here is that we simply let the cells attach to the surface and then just count the number of cells, either with or without shaking. So without shaking, yeah, the same number of cells will attach to the surface, but if you shake the plate, a lot of more cells were shaken off the surface. So fewer cells will stick to the surface, right? For the wild type as compared to the mutant. The mutant cells that stick to, they are resistant to the shaking. They still stick to the surface. So we think that somehow may explain that phenotype. But ultimately, we want to understand the cellular level, right? What happened? Why you have such a phenotype? And so very quickly, I like to show you this. So basically, we found that these mutant cells, they should increase the cortical dynamics. So here, just the video to show you this fact. So you can look at the wild type cells; they're very quiet. I mean, if they look at their cell membrane, they probably do not do any, like a dramatic dancing around. But if you look at the mutant cells, you can see their membrane is constantly, like this cell, for example, it's constantly throwing out something and then retracting. So it's very, very dynamic, okay? And so we can quantify these protrusions. You can see that, and the mutant cells, form many more of these protrusions. And this phenotype can be rescued by a transgene of YAP. So this is clearly a very specific phenotype. And so we can label the actin in these cells, right? And then look at actin specific, actin, what happened to actin. Again, we can show the movie to show you that you can see that actin here, like you can see that in these Y-type cells normally have very, like what I already showed you, these membranes very kind of quiet, if I use the word, but these mutant cells, they are constantly throwing out these extensions and then retract. And so I want to show you actually next slide, a detailed, a big blow-up view, a magnified view of a mutant cell to appreciate that. How these protrusions form relative to actin. So you can see that when these cells, when the cells throw out these extension, they are depleted of actin, okay? And then the actin flush into the extension and the cells retract the extension. So this is a very, very typical of what we call a bleb in mammalian cells. So we believe that loss of function of YAP somehow promote the bleb formation, in which the bleb is depleted of actin, and then actin comes in to retract the bleb, and that's how bleb can go out and come back. Okay, so this is very similar to what people normally observe in mammalian cell in this phenomenon called blebbing. And so as you know that blebbing is actually a very well-studied phenomenon, and there's a drug. It's called blebbing statin, right? Blebbing statin was initially discovered to inhibit the formation of bleb. That's why it's called blebbing statin. But later on, it's shown to be inhibitor of myosin. So what happened is that we can suppress, of course, the bleb formation in these mutant cells by simply treating with blebbing statin. You can show that the number of blebs, of course, is decreased. That's what we would expect. But perhaps more interestingly, we ask question: what happened to the aggregate morphology, right? Now what if you treat the YAP mutant cells, which makes this excessive bleb, right? Now you treat them with blebbing statin. Now they no longer make this bleb. We ask question: what happened to the aggregate morphology? And this is a very, very amazing phenotype we observe. That is, when you treat with blebbing statin, you not only rescue the bleb formation, which what you would expect, you actually rescue the aggregate morphology phenotype. That is, these guys, which the mutant cells which form this irregular shaped aggregate, now they form very round structure. And so that is really I don't think I want to go any further. I think I'm there. And so we have, of course, done some gene expression analysis. What is the take-home message here is that basically we saw that as consistent with our phenotype, we do not see any change in cell proliferation genes, but we see a lot of changes in genes related to actin binding, and a lot of membrane proteins. So I think this is kind of consistent with our observed phenotype. So anyway, I'd like to end here. This is really our take-home message. We think this pathway, especially this key regulator, it was initially evolved to regulate cytoskeleton and membrane dynamic in these early ancestors of metazoan, but only later they are adopted to regulate cell proliferation. And what is interesting is that in the mammalian system, actually the Hippo pathway is known to not only be regulated by cytoskeleton, it also actually regulates cytoskeleton as a downstream output. So it's kind of a complete regulatory loop. But somehow in this organism, we see a very profound regulation of YAP, right, on the cytoskeleton. And so that part is preserved, but it doesn't seem to have any role in cell proliferation. So we think the proliferation is only later co-opted during the evolution of multicellularity, and to make this, to integrate the cytoskeleton control together with cell proliferation control. So I think I'm going to stop here, and thank you very much for your attention. It's getting very, very late. I think I hope I did not take too much of your time. Thank you very much.
Great. Fascinating talk. Okay, there are several questions on the chat window. I'll take the pleasure to ask the first one. So when you showed the YAP response to the mechanical sensing and move from the cytoplasm into the nucleus, so this is also kind of like the cell response to outside stimuli, right? So this should be have something to do with the coYuki response as well, right?
Yes, yeah,
So
maybe
connected, maybe, yeah.
Mm-hmm.
So the YAP you used, is that overexpressed or endogenous?
The YAP, which is it's not overexpressed. It's a gene we can use to rescue the mutant. So it's a rescue construct, basically.
I see. Okay.
Yeah.
Oh, fascinating. Yeah.
Okay. Just because you're not going to moderate, I'm going to ask another question. Okay. Then Xing, I saw you raise your hand. You'll be the next. The question I have is, when I look at the movie, it looks at the mobility is reduced because aggregation seems slower in mutant than wild type. Have you guys checked whether chemotaxis or cell motility also impaired in mutants?
Yeah, first, we do not know of any chemotaxis, so we don't have like a DIPT, right? Okay. You can give them like some agent and show chemotaxis. So we don't have anything, and this organism is so poorly studied. I do not think Jonathan. I think Jonathan would attest to me that it does not affect mobility per se, because he could measure random movement, for example, right? He can track the cell. He does not believe there's any difference in what if I'm not using the right word, Brownian motion, for example. They're still joggle around in the same speed. So that I think that's what he concluded. Yeah.
Not related to DIPT. Initially, when I saw you in your aggregation, there's still cell proliferation. I thought, oh, perfect. It makes perfect sense because after DIPT aggregation, there's no cell de novo proliferation, right? They stop cell proliferation, then aggregation. But in the crazy organism you're studying,
yes,
after aggregation, they are proliferation. Initially, I thought, oh, massive proliferation. But then you notice aggregation, right? Then come to the question is, DIPT also aggregate together. Now, why the DIPT they don't need the YAP, but somehow the caps need the YAP?
I do not know.
No, it's so fascinating.
Let me give you a very I don't know the answer. There's one interesting observation which is very related to what we saw. So especially like I end here on the last slide. So we like to entertain this very crazy hypothesis. If you look at this step, right, the second step, the third step, which we think YAP is required. So we think maybe this pathway, the YAP, initially evolved to allow 3D structure formation. So basically, the idea that anything on Earth, you have to resist in a solid environment, like in any environment, you have to resist gravity, for example, right? So you need to have some kind of 3D structure in order to perform physiological function. So I think this is the idea, and actually related to this, there was the Maduka fish mutant of YAP, which was published by a Japanese group several years ago. They show that these Maduka fish, so of course the normal fishes have certain 3D shape, rigidity, right? These mutant, they are just like this. They're just like floppy, and they're alive. I mean, they're just like soft. Just like the people who are missing like calcium or whatever, that's the idea. So it's very interesting. I think there's some parallel there, but of course they don't know the mechanism. But I think maybe intuitively, I think this may be required for the initial evolution. You need to have 3D structure so that they can move together to do something. So.
Okay, Xing.
Hey GJ, yeah, fantastic talk.
Thank you.
Hi DJ. Yeah. So hey, so I wonder, if this regulating proliferation and anti-apoptosis is kind of a gained function in multicellular organism. If you compare the sequence of the YAP in unicellular organism versus multicellular organism, would you can you uncouple these two functions?
You mean in higher organism?
Yeah, because you are talking about in this unicellular organism, the main function regulating cytoskeleton function, right?
Yes,
While
yes.
in a multicellular higher order organism, they regulate proliferation, they anti-apoptosis. So I just wonder if these two functions in, if it's a gate function during evolution, are these two uncoupled?
Oh yeah, so maybe I did not make myself clear. So the cytoskeleton function still preserved in the mammalian and fly species, okay, in the higher organism, and
Yeah.
so it just added this proliferation control to this old machinery. That's my
I wonder, if you compare the sequence, can you see like two different domains or
No, I don't think so. Let me tell you why. Because I did that experiment like 10 years ago, right? When we overexpress the Capsospora yorki in fly eye, it drives cell proliferation. So it's not at the protein level. These proteins, they recognize the DNA sequence like the fly homolog. And so the biochemically, I don't think they are different. It must be the wiring of the genome, the epigenetic. God knows. I don't know. I mean, I think that there's something else at a higher level. Yeah.
So in that way, it seems it's context dependent, right? So, for example, it's tightly controlled. During development and during regeneration, it's high, but during normal homeostasis, it has to be suppressed. So that raises another question. If you change the environment, what would they respond? For example, if you have the cells in the young animal to the older animal, or from the regenerative tissue to the normal uninjured tissue, would they change the behavior?
I
different mosaic analysis.
I think those are different experiments, but I do think those are relevant experiments. If there's good system to do that, I think those are
more like a transplantation. Like take out of the context and put in a normally non-responsive environment. Would they lose the ability to proliferate, or they will keep doing that like totally cell autonomously?
So related to that, we do know that YAP and yorki is involved in cell competition, in which as you know, it's a phenomenon that is involves cell comparing to their neighbors. So it's not a completely cell autonomous for sure. So they do play some function in a kind of social environment for sure. Yeah, even in flies.
Yeah. Great. Yeah, fantastic. Thank you.
Okay, I'm gonna read one more question from the chat. So Zhao Zhu was asking, does UK also undergoes mechanosensitive translocation to nucleus in this single cell organism?
Yeah, that's a great question. So we haven't been able to do that experiment so far. So we would love to do that. So the only experiment we kind of did, that, I mean, not even exactly to that question, but is related to that question, is that we do know that this particular yorki has the same phosphorylation sites, okay, as in their fly or mammalian counterparts. So we could mutate this Hippo responsive phosphorylation site, and they would go to the nucleus, okay? So that we know. But yeah, we have no idea about what the upstream. If you pay attention to my talk, you see that so far I didn't show you anything about the Hippo phenotype or the worst phenotype and all of that. So we actually have those mutants, but their phenotypes are less straightforward. So we are trying to look at them more carefully. But clearly they do not have proliferation phenotype, that we know. So we are trying to characterize their cytoskeleton phenotype to try to make sense whether they are the opposite of yorki or not. Because as you know, when you have the cytoskeleton phenotype, sometimes it's very hard to show, opposite of cytoskeleton. Let's say you have more dynamic cytoskeleton, what would be the opposite? I sometimes it's just hard. So I think we still have some questions there. But yeah, we would love to, if you have some way to, I know you think about mechano transduction every day. So if you have some way to suggest for us to manipulate mechanic force in these cells, I'm sure we would love to take a look. We have all the tools, we can do it. Yeah.
Okay, so another question from Yonghe, I thought is really interesting. He was asking, because YAP you're showing is induced by a mechanical force. However, in cirrhosis, the liver is very stiff than normal soft liver. So could this may also potentially induced by YAP?
Which I think it would be easier for me to read cirrhosis question, right?
Yes.
Okay.
So he was asking, in the cirrhosis, the liver is much more stiffer
Yes.
than the normal soft tissues. So you think
Yes, YAP is inducing cirrhosis. You're right.
Yeah.
Yes, yes, yes.
That's
They
cool.
always, so actually, liver cirrhosis is a major inducer of YAP and liver cancer as well. So I think this is a very relevant, actually consistent with this whole idea of stiffness. You know, your cells more stiff, you will turn on YAP. Okay. Actually, by the way, I just want to leave with you. I know I'm getting. I forgot to tell you the slide. So I'm just trying to promote this organism. So with my last slide, so this organism, I think it's a wonderful genetic model. It's not only study Hippo pathway, but to study many other interesting questions you might be thinking about. Okay, this organism, it has integrin, it has P53. Okay, so you have P53, it has MYC. You can look at this diagram, find your favorite gene. Okay, so I'm leaving it here. So if you're interested in studying anything, and cadherin, for example, here, so now we have the tool to overexpression and lockout. So if any of you want to ask a question, like for example, this is the simplest organism in which you can find P53. Okay, so we are actually very interested in learning what P53 is doing, because I think those are the kind of question you can really answer in this kind of non-canonical model.
Okay, thank you, DJ. We probably should, if you have more questions, you want to ask DJ, if DJ can stay on a little bit.
I will stay on for a few minutes.
That's fantastic. Yeah, could continue. We can maybe in the break room. So thank